after 2 minutes, how long did the student exercise for? Right, so we can see what it's asking you to do here is identify the starting time. So this point here is 0. That is where they start exercising. We know that when they stop exercising, that their heart rate is going to start to come down, and the point that that happens is here on the graph. Now if we count how many years that is, we've got 1, 2, 3, 4, 5, 6. So it is 6, not years, sorry, it's 6 minutes, not years. Okay, so if we circle 6 minutes, we can see on the graph why that is 6 minutes there. Okay, so the link that you need to make here was that when the heart rate comes down, that's when they've stopped exercising. So it's returning to a normal beats per minute. Okay, we'll move up. Okay, now 3C part 2 says aerobic respiration uses oxygen. To take in extra oxygen during the exercise, the student's breathing changed. Give 2 ways breathing changes during exercise, and that is for 2 marks. So for the 2 marks then, you needed to say for what your first one, that there is an increase in breathing rate. So if you need to get more oxygen into your body, because your cells need the extra oxygen for respiration, you are going to start to breathe a lot faster. Okay, so you get out of breath, you start to breathe a lot faster. The second point that you would need to mention is that the breaths actually get deeper. So we're taking in more oxygen with each breath. So it's nice and straightforward there for 2 marks. The mark scheme would also allow it if you said that the person would have shorter breaths or heavier breaths. Any of those are okay. And if we move over onto the next page. A little bit blurry this one, but we can still read it. So it says, when a person is exercising hard, oxygen cannot reach the muscles fast enough. This means aerobic respiration cannot provide all the energy needed. Complete the sentence. So it says, during hard exercise, extra energy is released by missing word and then respiration. Our missing word there is anaerobic. So that takes place without oxygen. So it has to be anaerobic. You need to make sure as well, folks, that your spelling of those 2 words is spot on. Otherwise, the examiner, if he's confused about what you've wrote, he will not give you the mark. So make sure that your spelling is clear for that one. Okay, next page. Okay, so we've got question 4 now. So protease enzymes digest proteins, and it's now saying what type of substance is produced when protein is digested. So it's a basic knowledge and understanding question. The answer for this is amino acids. So when we break down large protein molecules into smaller molecules during digestion, we get amino acids. Correct that spelling there. So amino acids. Okay. Okay, so if we move down, it says a student investigated the digestion of protein by protease. Egg white is a protein. Boiling changes egg white from a clear liquid to a white solid. The students did the following. So step 1, put 10 centimetres cubed of liquid egg into a measuring cylinder. Put the measuring cylinder into a boiling tube until the egg white went solid. And then he put 5 centimetres cubed of protease solution into a test tube. So the protease is your enzyme that's been added. Anything that ends in ase is an enzyme. But both the measuring cylinders and the test tube into a water bath at 15 degrees Celsius for 10 minutes. So he's putting them in there together for 10 minutes. And then he added the protease to the solid egg white after the 10 minutes were up, and recorded the volume of solid egg white after 24 hours. So it is important for these questions to read through the details and pick out the key things. So if he's putting them in it for a certain amount of time, start to think about why that might be. Okay, and then we've got our results just here. So we've got step 4 there, which is where he's got the egg white and the protease in the water bath for 10 minutes, and then we've got the results after 24 hours in step 6 here. Okay, so we'll turn over and we'll refer back to this page in a second. Okay, so for this question it says, in step 4, why were the solid egg white and protease solution put into the water bath for 10 minutes before being added together? This was basically to make sure they were at the same temperature. Okay. If they weren't at the same temperature, it wouldn't have been a fair test. So it has to give it time to allow that to happen. And then B, part 2, calculate the volume of solid egg white digested after 24 hours at 15 degrees Celsius. And we've got to use figure 14 to work out the answer. So what we'll do is we'll bring that in now. Okay, so the diagram should now be on the screen from the previous page, and it shows the results before and after the experiment. So if we look here, we've got 10 centimetres cubed of our egg white, so that's a solid egg white, and then after 24 hours it has decreased to 7 centimetres cubed. So we know over the 24 hours it's gone from 10 to 7, and it's asking us to calculate the volume of egg white that has been digested. So that is the volume that is no longer there. So what we need to do is 10 for our starting amount, minus 7, which is the finishing amount, and that gives us 3 centimetres cubed for our answer. So that is a 1 marker. Quite nice and simple, that one. A lot of people were getting mixed up, and they were just putting down 7 instead. They were just reading it straight off the actual results that were down here. You've got to look into it in a bit more detail. So look at the starting amount, finishing amount, what's happened to that amount that's disappeared. It has been digested. Okay, next page. Okay, so other students repeated the investigation at different temperatures using 20 centimetres cubed of liquid egg white. The students used the same concentration of protease solution for each temperature, and table 2 shows those results. So we'll make this a little bit bigger so we can see it. So we have got at 10 degrees Celsius, we've got 2 centimetres cubed being digested. At 20, we've got 4. 30, we've got 8. 40 is 16, and then 50 is 0. Okay, so the question says, describe the relationship between temperature and the volume of egg white digested at temperatures from 10 to 40 degrees Celsius. Okay, so it has to be from 10 to 40. You are not looking at this one here. You do not have to look at 50. Those results do not matter. The question's not asking you to look at them. Okay, so if we look at the results, the relationship, so we can see as the temperature increases, so it goes from 10 to 20, 30 to 40, the volume of egg white that's digested starts to increase. So it starts at 2, 4, 8 and 16. Okay, so there was 2 marks. So your first mark is for saying as the temperature increases, so does the volume of egg white digested. Okay, so that was for 1 mark. Okay, and your second mark that you get for that question is for saying that the amount of egg white doubles. So we can see as it goes up from 10 to 20, it doubles from 2 to 4, and from 20 to 30 it doubles again from 4 to 8. So it doubles in amount each 10 degree rise. So you had to spot the pattern in the data there with that one, folks, which most people did quite successfully. Okay, right, so if we move on to the next page, I think we finished with that page, just double check. Group those. Yep, so we'll move on to the next page. Okay, so for this one, student 1 wrote a conclusion. Protease works best at 40 degrees Celsius. This conclusion may not be accurate. Suggest how the investigation should be changed so that student 1 could find a more accurate value for the best temperature. Okay, so what you need to do for this one then, you've got to look back at the results. So we're going to bring the table back in and we're going to look at that now. Okay, so the table should now be on your screen, and we can see that the temperature only goes up in 10 degrees Celsius intervals. Now, what the examiner is asking you to look for here, folks, is that we could improve the investigation by doing repeats at other temperatures. So we could repeat the experiment at other temperatures. and it might just want you to give some examples for that. So you could say repeats at 5 degrees Celsius intervals. So repeats at 20, 25, 30, and so on. And that will allow us to gain a bigger picture as to what temperature the enzyme works best at. Okay, so student 2, let's look at theirs. So it says student 2 wrote a different conclusion. At 50 degrees Celsius, protease is killed by the high temperature. Why is this conclusion written by student 2 not correct? Really basically, it is saying that an enzyme is being killed, and an enzyme is not a living creature. So the enzyme is not killed because it's not alive, it's not a living thing. Ways for them to improve the conclusion for the next mark is by saying that the enzyme is denatured instead. Any description of denaturing, so that is essentially where the active site of the enzyme changes shape due to a change in temperature, or it could be a change in pH as well. But it's something that makes the active site of the enzyme not work. So by that, just show you a quick diagram. What we mean is, let's say this is an enzyme, and we've got here, this bit here is the active site. So we'll just put active site. And this is essentially a huge network of proteins. So this is made of protein. The way that an enzyme works is that a substrate actually fits into this slot, and that substrate then gets broken down into smaller bits. So it might break it down into something that looks like this and this. Okay, so that's the way an enzyme works. So this could be your long chain protein, it gets broken down, and these could be your amino acids. What actually happens when the enzyme is being denatured, I'll try and show you that now, just move these around a little bit. So yeah, as I was saying, what happens when the enzyme is denatured, it changes the structure of the active site, so it may now look like this instead. So it actually changes the structure of the active site so that that substrate can no longer fit in there and the reaction can't take place. So that would mean that there was no reaction. Okay, so that was what it was trying to get you to think about. So it's understanding the key language here. Denaturing was a word that you had to mention or had to describe in order to get the marks for this question. Okay, so we'll go on to the next page now. Okay, so we're on question 5, and it's talking about parts of a cell. Nice and straightforward, just a simple knowledge question. So it says, draw one line from each part of the cell to the correct function of the plant. Okay, so cell wall. The job of the cell wall in this scenario, the best function that you can match it to, is to strengthen the cell. So it provides support and strength for plant cells. The chloroplast, most accurate one that we can match it to, is it absorbs light energy to make food. So it's actually the chlorophyll that are inside those chloroplasts that absorb that light energy. And then the nucleus controls the activities of the cell. So that was a really nice free marker, one that we shouldn't be losing marks on really. So make sure if you did make any mistakes, you go over cell structure and what the jobs are of those different parts, because this is basic key stage 3 knowledge. This is key stage 3 cells knowledge.